We are live, sir.
All right, very good morning, everyone. All the students who are joining us for MOOC live session 2 for the Bioengineering course and for the Interactomics course. Welcome to today's live session. I think you have been studying various proteomic technologies, and we have given you different flavor of how these technologies can be used for different applications. Today, what we are going to we are going to focus on one specific proteomic technologies, mass spectrometry, which I'm sure by now you have recognized that is very important technique and tool which has given a power to the proteomic scientist. And now we are able to investigate proteome and proteome scale problems at the very unprecedented level, which was not possible earlier. And therefore, today's session is going to be more thematic on looking at mass spectrometry-based proteomics. With me, we have also joined various TAs of the course who are going to take you directly from the lab and show you different instruments and different technologies. I'm currently as a visiting professor working in University of California, San Francisco on my sabbatical and conducting this session in the night time here from California. But you will still get the flavor of the mass spectrometers and the lab directly from my IIT Bombay lab, and TAs are going to show you different applications and different instrument configurations. So, as you have studied in these courses, that proteomics started with the conventional two-dimensional gel electrophoresis or gel-based proteomics, and that was kind of a very simple and important technique which helps scientists to investigate proteome at a simplified level, not at the very deep and very complex proteome, but looking at maybe a few hundred to thousand spots or proteins could be analyzed. However, the process of doing the two-dimensional separation was quite complex. You are preparing the samples, you are doing the isoelectric focusing, you are doing the SDS page for second dimension separation, and only then you can now look at each tiny spots on the gel, which can give you some idea about the protein spot. Then you excise the band or the spot and do in-gel digestion and run in the mass spec to identify what that protein is. So the entire procedure was a lot of cumbersome steps were involved in looking for this kind of proteome scale investigations. So one thing which was kind of the major change which happened was the advent of shotgun proteomics and a concept known as MUDPIT, multiple dimension protein identification technology, that came forward, which really showed that it is possible to avoid the gels and directly one could prepare the samples and inject the samples in the mass spectrometer for direct protein identification. So that was kind of a big change, moving beyond the gel-based proteomics and now moving to gel-free mass spectrometry-based proteomics. So the shotgun proteomics essentially, as you can see in the slide here, uh, looks for simplifying the proteome scale investigations. Take the your cell lysate or tissue or any biological samples, prepare the lysate, and now you are going to perform the urea buffer exchange, reduction and alkylation steps, followed by digestion with the trypsin enzyme. And then those digested peptides, which also have the buffer component and the salt, you want to clean them up and a process on a desalting of these zip tips. And then you can run the samples either with the MS1 or do MSMS analysis for the peptide identification, protein identification. And then of course the complex data being generated, which you need to search against different databases. So this is what a general mass spectrometry based proteomics, also known as shotgun proteomics, is now simplified, and you can see in these steps you can do this kind of work very with great ease. Now the the maximum output, one would say, of looking at these mass spectrometry technologies was realized in 2014 and 2015 when we had the first time the draft human proteome maps were published based on the mass spectrometry-based identification of all the human protein. So idea was like, since the Human Genome Project got completed in 2001, uh, can we, replicate the same success and try to now identify all the gene coding proteins, but whether we have experimental evidence for that. So in this light, we, scientists looked into entire human proteome and try to provide evidence that the protein do exist in the human body. Uh, but still the success was only 70% or so of looking at the entire draft human proteome map. Uh, Further, this work was continued, and during the COVID time, in fact, in 2020 November, a big team of Human Proteome Organization and Human Proteome Project, including our group from IIT Bombay, a manuscript was published known as the High Stringency Blueprint of the Human Proteome in Nature Communication, which revealed 90.5% or 91% of the human proteome with very high stringent criteria one could now decipher. Since now we are talking about high stringency criteria, and since you are the students of these course of Interactomics and Bioengineering, where you have been exposed to some of these technologies, so let me briefly mention that while mass spectrometers can generate various peptide sequences, and you can look at large number of protein identification from that, but as a part of protein identification, you need to be very stringent of using some cutoff and the filters and statistics to ensure that your search which you're going to have based on these peptides is confident enough to identify the protein. And I think that's where the FDR 1% FDR of looking at the cutoff filter, looking at least two unique peptides of a given protein, and peptides are non-nested, and the length of peptides are 9 or so amino acid, were used to build this type of draft human proteome map. Uh, Further, then the technologies like Orbitrap fusion mass spectrometers, they came forward, which has really expanded our power of looking at proteome coverage, proteome analysis. This is one of the tribrid mass spectrometry based technology, which is having the quadrupoles and Orbitrap and ion trap, and one could analyze the the proteome after coming from the quadrupoles, either in the Orbitrap or in ion trap, or use both of these mass analyzers. So this is the tribrid mass spectrometer, which has really given us a lot of power of looking at proteome-wide investigations. There are many other mass spectrometers currently being evolved, which are now equally powerful and trying to look at different type of proteomic analysis. But the Orbitrap technology, and especially Orbitrap fusion, was the first in the series which gave us that kind of power. So one could look at different type of applications of proteomics using the mass spectrometer. It could be based on the whether you have your control and disease condition, which you want to label with different type of tags, and these are isobaric tags known as tandem mass tag TMT, or iTrag, isobaric tag for relative and absolute quantification, where you have different reporter ions, in this case 126 to 131 for the 6-plex of the sample. So let's say you have 3 control and 3 treatment conditions where the peptides have been extracted from the complex samples, and then those are being labeled with these reporter ions. These are isobaric tags, and then you mix them all together, and then you are going to run into LC-MS to look for the fragmentation pattern and the reporter ions to use for the calculation of these intensities for the fold changes of control versus disease condition. In a very nutshell, I'm sure you have seen some of these concepts, and we'll be happy to talk more if you have more detail which you need. But I'm not giving a theoretical lecture today. I want to save time for showing you these things directly in the lab. So, but still, I'm trying to give you a broad overview so that you are prepared to see in the lab what you can do. Then additionally, we had the label-free quantification technologies which developed when you don't label the protein, but rather you are running every individual samples at the proteome-wide scale completely, and use the spectral count of the peptide to measure the changes from the control versus disease. or now a newer approach. So these two were DDA-based method, data-dependent acquisition, whereas now a new method known as DIA, data-independent acquisition, is also evolving, which is evolving very rapidly now. Given that we do see some missing values and the some errors in the measurements in the DDA-based method, DIA methods are becoming more powerful, where you are trying to look at each of the mass window with higher resolution and trying to build the library, and then you are going to map your proteome samples on that library. So these are, in a nutshell, various quantitative proteomic technologies which are currently being used in the field of proteomics. Whatever sample you are working on, whatever biological or clinical project you are interested to work on, these techniques are going to be equally powerful for your applications. So during the COVID-19 time, we at IIT Bombay and many other labs worldwide utilize the power of mass spectrometers to look at various type of problems. To look at, first of all, virus diagnosis was the challenge, which was overcome by RT-PCR. But simultaneously, scientists also try to look at whether we can directly identify the peptides of the virus rather than trying to amplify the cDNA and do the RT-PCR. Can we just directly look at the presence or absence of these viral peptides in the samples, which will have probably more impact. Also looking at from the plasma and nasopharyngeal swab samples, looking at the deep proteome or the entire protein which gets changed because of the viral interaction, as well as looking at the host and virus interaction map and the immune response. All of these were different type of technologies which were being used, especially mass spectrometer, but also in the protein arrays were used to investigate this problem. So of course, today's focus is going to be on the mass spectrometry research, but also we will try to keep in mind COVID-19 as a case study. Uh, of course, we are going to apply the same instrumentation and same workflow for any application for that matter, but you can think about this as a case study for using the mass spectrometers. So when we were trying to investigate the COVID-19 problem, uh, first we were getting the nasopharyngeal swab samples from the hospitals, where it was only the idea was, can we take the leftover samples of these patients, and very generously our various clinician partners from Kasturba Hospital and Jaslok Hospital, especially Dr. Jayanti Shastri and late Dr. Om Srivastava and many other clinicians, they collaborated with us and helped us to look at various SOP which we could optimize for the viral peptide extraction from these nasopharyngeal swab samples. Of course, many challenges were there, which I'd not like to talk, but idea was, can we inactivate virus? At the same time, can we also look at the viral peptides using mass spectrometry-based methods? And once we do that, then can we further take our investigations forward and develop the simplified test known as the multiple reaction monitoring based assays. So I'm just showing you here one of the sample prep which we did for the nasopharyngeal swab sample, and of course, as you can see, the challenges were immense for working on these kind of clinical samples, given the safety considerations, and you have to inactivate virus first. So we tried different organic solvents from ethanol, acetone, isopropanol, and try to ensure that we are first inactivating virus. We are incubating for a few hours with these organic solvents and precipitate the protein out. And then further, we would like to dissolve that protein pellet into our lysis buffer, and followed by the digestion of the peptides. So this was the kind of workflow which we followed in this kind of sample preparation. But let's say you have now prepared the lysate and you want to proceed for the the next steps forward. So in proteomics, what you will realize that we have to do at each step, we have to make sure that we are in the right direction. So we do a lot of the quality control checks, right? Whether our protein which we are extracting is having sufficient quantity to proceed for the digestion of the peptides. So that is the first consideration. So that's where I think different type of protein quantification methods, including Bradford and BCA methods, are being used. In our first direct live session from the lab, we are going to show you the protein quantification. And then once you know that you got 100 microgram protein or more, and you want to digest 50 microgram protein to get sufficient peptides, then you have to do the peptide quantification and also ensure that you got sufficient amount of peptides from the given sample. So these are the kind of thing which we do. So let me introduce here TAs who are going to take this session, and they are going to walk you through the steps briefly about protein quantification and peptide quantification. So Avinash, Ankit, and TAs, over to you. You are not audible. Also, I think let me stop sharing so that your window can be made.
Yes, good morning, everyone. So I'll be demonstrating how to do the protein quantification. So as I mentioned that we first need to quantify that protein, how much protein we'll be digesting and how much protein is present in the unknown sample, so that we can take that equivalent amount of volume of protein for digestion. So for that, what we do is that we take Bradford assay to form the protein quantification. So as you know that the Bradford, when it with the amount of the protein, it changes its color from brown to blue, which we measure its absorbance, and then we can find out the concentration. So first, what we do is that we make a standard curve using a known protein concentration of PSA. So here you can see that we have taken a blank without any protein concentration. Then we have taken 2 microgram of protein, and from here we have made the standard curve till 6, 8, 10, 12 till 12 microgram of the proteins. So we have taken, so with the PSA of the initial concentration of 1 mg per ml, that is 1 microgram per microliter. So we have taken the concentrations like 2 microliter, 4 microliter, 6 microliter to make the standard curve. And then we have taken the unknown samples to microliter and added the Bradford. So you can see that there is a change in color. So from here, after the standard curve, we would try to understand the concentration, what is amount of protein is present in this unknown sample. So after we keep the Bradford, we incubate it for 15 minutes, but we have already done that. And after the incubation, so we put it in a 96 well plate. Like here, we after adding 1 ml of the Bradford, we take 200 microliter from that mixture and add in this 96 well plate, and we place it in a UV spectrophotometer like multi-scan. So that's what we are taking, and then what we do is that we make the plate layout, where you can see that we have taken the 96 well plate and we have taken blank and the standard concentrations and then the unknown. And then we do is that we set the protocol to measure the absorbance at 595 nanometers. And also we perform the blank subtraction and we start take the reading for this. So we have taken one reading. So after the reading is done, so here we can see that there comes the graph. So from here we have taken the known concentrations 2, 4, 6, 8, 10, 12, 14. So we prepare a standard curve like this, where you can see that the intercept is set at 0 and the R square is coming 0.992. So after this curve, from here with the help of this equation, we can calculate that with the absorbance what we get for the unknown sample. So we can calculate that what is the actual concentration of the sample, and after we get the actual concentration, then we can go for further processing and digestion with technique.
So once we have quantified the sample, and once we know that how much protein is there in the sample, we can accurately then move forward towards digestion of the protein. Now, digestion of the protein is very important for mass spec-based analysis, because an improper digestion will not be accurate. So to make sure the protein is accurately digested, so we prepare the protein very well. So by preparation of the protein, while preparing the protein, we have to make sure that all the disulfide bonds are properly protein in the protein, and so that the trypsin has accessible to the site. Now, so the first step of digestion, we take the sample. So this is the sample. We always try to keep it in the ice. We have measured the protein concentration, and according to that, we have prepared the reaction volume. Now we are first going to add a reducing agent. So here, reducing agent is important for reducing the disulfide bonds that are present inside the proteins and in between the proteins. So we are using TCEP. So it's tris carboxyethyl phosphine. So it will break the disulfide bonds. So we will first add the TCEP, and then we will incubate the samples for 1 hour into 37 degrees centigrade so that the digestion is proper and complete. Now, once it is done, we will incubate the sample in this incubated shaker chamber for 1 hour. Now, once that is done, the next step will be the alkylation. Why the alkylation step is necessary? Because so the disulfide bond that has been broken, so that they cannot reform. So we will use isoacetamide for the alkylation process. So isoacetamide we will add in the sample once again, and we will so all these steps are the preparatory steps for the protein digestion. We'll add the isoacetamide, and we will incubate the entire thing in dark, because isoacetamide is once we have done that, so we will incubate with isoacetamide in that for 15 minutes. So once that is done, now our protein is prepared for digestion. We will make sure we will add a buffer into it, which is called a digestion buffer. So digestion buffer is required to maintain the pH of the reaction mixture. So this is important because the trypsin, which is an tryptic digestion, which is an enzymatic process, so it requires a certain amount of pH to perform the reaction. So these are the pH strips. So before adding the trypsin, before adding the digestion, we make sure that the pH of the solution is nicely maintained. So we will take the sample reaction mixture, and we will add the digestion buffer into it, and we will try to check the pH. So this is the digestion buffer I'm adding to the sample, and try to check the pH. So if you can see, the pH is equivalent to pH 8. Now your sample is completely prepared. Now you can add trypsin and perform the overnight digestion. So at the last step of digestion, add trypsin to the reaction mixture, and we will keep it in 37 degree overnight for the digestion. We'll use the same incubator shaker chamber for the overnight digestion. We generally keep 16 to 18 hours here. Then once we are done that, so we need to concentrate the peptides. So once it is done, we will in a feedback chamber. So this is the instrument where we dry and concentrate our peptides. So if I keep the sample over here, it will take a couple of hours to the drying of the peptides. So once it is done, the next step of sample submission would be now, if you know that when we digest the peptides, we use a lot of buffer, and that has a lot of salts in it. So before submitting the sample, we need to purify this digested peptides. So we use this particular C18 column material. We pass the sample through this column material to purify the peptides. Once that is done, we quantify the peptides after purification. So this is the plate that we use for peptide quantification. This is a mu drop, thermoscientific mu drop plate we use for peptide quantification. So once we have digested and concentrated peptides, we dissolve those peptides into acid, which is 0.1% formic acid, which is an excellent solution for dissolving all type of peptides. So we first dissolve the peptides, purified peptides, and then go for one round of peptide quantification. And then if that we get accurate amount of peptides in this quantification, we submit the sample to MS.
All right. Thank you very much, Amrita, for giving this demonstration, as well as Ankit. I think all the students would have realized that, before you actually take the samples forward on the mass spectrometers, you have to do several steps of quality control check. And in a very nutshell, you have got the glimpse of these processes of doing protein quantification, digestion, and peptide quantification. So at IIT Bombay, we have this mass spectrometry facility, which is a national proteomics facility for research and training, which is equipped with various instrumentation and technology. Like what you see here is Orbitrap Fusion, which I spoke about, a tribrid mass spectrometer. This is the LCMSMS Q executive mass spectrometer, which is a hybrid mass spectrometer, and this is a triple quadrupole based TSQ Altis. So different instruments can be used for various applications in mass spec and in general biology for various application. But we primarily use for the deep proteome analysis, or the looking at the entire proteome coverage, the one of the best highest resolution mass spectrometer, which is Fusion, shown in the center here. And while Q is equally good in doing the proteomics, but we primarily focus on using this setup for metabolomics based analysis. And ARTIS is being used for targeted proteomics, targeted metabolomics kind of applications. So being a national facility and a resource which all of you can utilize as well, we'll be happy if if you as a user would like to come here and provide your samples or come for training. We'll be happy to facilitate that as well. So in the next series of live sessions, we are going to take these various instruments of mass spectrometers and give you the live demo from the lab, uh, where you will see like these peptides which have been prepared. Now, how to take these peptide samples, put in the auto sampler in the LC, and because the first you want to separate the complex peptide mixture what you have with the simplified fractions, and you would like to do that in the live online manner using the nano LC, followed by series of injections happening directly into the mass spectrometer, so that the complex peptide fractions could be what you have in different elute from the LC could be further analyzed with the mass spectrometer. Let me now request TS to give you the tour of mass spectrometry facility at IIT Bombay, where first Dr. Arghya Banerjee is going to show you the Orbitrap Fusion mass spectrometer. All right, so Arghya, over to you.
So, hello all, welcome. Like I will be showing you the demo of the mass spec. So what you are seeing is the cutting edge technology, which is the Orbitrap technology, and this is the mass spectrometry, that is Orbitrap Fusion. It is a tri-hybrid mass spectrometer which has two mass analyzer. One is ion trap and one is the Orbitrap, and yes, with the quadrupole also. So that is why it is called as a tri-hybrid. We do extensive sensitive study at very microliter level, mul level in this instrument. So that is why we use this easy nano source LC. Okay. So this is specialized for carrying out the protein based experiment, mass spectrometry experiment. It can withstand up to 1200 bars of pressure. And second thing is, the most important thing is that it has a flow rate of maximum to 2 microliter and minimum like we can channelize. So here you can see the parameters which are there. Like here you can see the parameters which are there. This is the software interface of the Nano LC. So now just we will be inserting the samples. So we will be ejecting the tray.
this is how the solvent tray looks like. So these are the solvent reservoirs which we have to fill every day, and these are the tray. Now what I will do, I will keep the peptide samples that have been prepared. I'm keeping the peptide samples that has been prepared, and again I am ejecting it. Means again I am placing it. So there is a matrix. Now you have to keep in mind, you always have to do a instrument checkup. Second thing is a QC control, so that it will tell about day-to-day instrument variability. So once that is done, the solvent will be coming from this tube, like the peptide samples will be coming from this tube. It will entering into first one pre-column. What is the function of this pre-column? This small particle. It is all C18 column. So the function of this pre-column is to avoid any sort of salt or any matters which is being taken out from the sample solvents. And once that is done, the clean solvent is released. What you are seeing, this black circle thing, this is the main analytical column. This analytical column will now inject the solvents into the ion trap, the ITC, isothermal calibration tube, which you can see in the camera, which is a bit hazy, but you can see like this is the sample source. And after this, the samples will be injecting insert. So now the thing is that you can see check out in the main screen. The samples will be eluting on the basis of the RT and M by N. Like this is a time based as well as the protein abundance based. So yeah, so now you can see the samples will be eluting. The samples will be eluting at a particular on the basis of the relative abundance and the time. And we do monitor the individual program. So suppose if I can open a sequence. So this is how the sequence interface looks like. If we open a sequence, so here you can see these are the sequence, and we generally run a quality samples, the PRMs. After that, we do a traditional in routine sample. So this is how a chromatogram looks like. This is the relative abundance and this is the time abundance. You can check the individual peaks of these chromatograms. So you just go and zoom and you check the individual peaks, how the individual peaks are coming. So these are responsible for the each of the chromatogram, and you can see their MSMS fragmentation at this bottom column of the interface. So this is one of the most advanced technology in the field of mass spectrometry, which we are using, and this can suffice like the coverage. It has a high coverage, high resolution, and high detection. So any kind of study, either it is a label-based study or it's a label-free study, can be conducted. Now I would like to request Professor Sanjeeva to give the take the forward.
All right. Thank you, Argyo. And so now I think students have got a live glimpse of the instrumentation. Uh, but this process of acquiring data of mass spec required some instrument method setup. So let me play this video clip of Argyo on showing the instrument method setup.
So once you have injected the peptide, like you have placed your peptide into the autosampler, the next point comes into the method development. So you have to develop a method for your run. So where we are using orbitrap fusion for our run and thermo nano LC. Now these are the important parameters which you need to know. First is the sample pickup and loading. Then is the sample loading. What is the maximum pressure that can be maintained? In case of gradient, here comes the main funda, like how much time of gradient you are using. Generally, my peptides are hydrophobic. Whatever peptides we are using is hydrophobic. I am using a C18 column. So basic logic is I am using two solvents. One is hydrophilic and the other is hydrophobic. That is A and B. My solvent A is hydrophilic and my solvent B is hydrophobic. When I am gradually increasing the percentage of my hydrophobic solvent, so whatever the hydrophilic peptides are there will be eluted first, followed by hydrophobic peptides. Okay. So this is the logic. We are generally increasing the hydrophobicity, and those which are more of the hydrophobic will be eluted at the latter stage. So here we are using an 120 minutes gradient, means 2 hours gradient, where my solvent B or my hydrophobic solvent, hydrophobic polar solvent is increased from 5% to 90%. So whatever the my peptides will be coming will be all eluted. So once the gradient has been set down, you will go to the pre-analytical and analytical column. These are basically the C18 columns. The maximum volumes which can be included for the pre-column is around 10 microliter, and that of the analytical column is around 8 microliter. And the maximum pressure which it can adjust, like when the column pressure is existing 750, you will know that there is some problem. You have lot of unwanted materials in your solvent, so you have to in the peptide solution, so you have to again clean it. So max pressure is around 750 bar, and this is the 750 bar. This is the auto sampler position, where you can see like what samples are placed and what position. And this is the easy nano spray, like from here the nozzle will be sprayed. Okay, so now you move on to come to the main machine, like the Orbitrap mass spectrometer. In the Orbitrap mass spectrometers, you will have different parameters such as global parameters. In the global parameters, in the application mode, you can decide like whether you are analyzing peptide, small molecule, or an intact protein. We are generally using a peptide, so we have selected peptide. And the method duration, you have to sync it here. You have to make it 120 because we are using 120 2 hours gradient. Okay, this is method duration in minutes. You go to the scan parameters. In the scan parameters, actually these are the basic parameters which are generally used. In the scan parameters, there are various options starting from the MS, whether you want to do a full scan or selected ion monitoring, whether you want to do MS mode or DR, which is used for the targeted workflow, and various other sub parameters which can be used, such as the target mass trigger. This can be used in the DR, means data independent acquisition. So in the MS Orbitrap mode, if you come to the MS Orbitrap mode, whatever the default chart state is set to 1. Here need to define the postulates. Okay, here we are using a resolution, Orbitrap resolution of 60,000, but it is capable of going to 5 lakhs. You can see around 2.4 lakhs. Here it is 5 lakhs of resolution. The scan range, what we are using is from 375 to 1800 nanometer. The scan range what you're using from the 375 to 170 nanometer. The RS range is 60. The AGC acquired target is 4 point into e to the power of 5. Maximum injection time is 50 millisecond. We have set to 50 millisecond. You can optimize it according to your protocol. And finally, the polarity, we are observing the positive mode. But in case of metabolomics experiment, you can go for positive and negative both. The monoisotropic peak determination is kept to the peptide rather than the small molecules or the proteins, because we are analyzing mainly the peptides. The intensity, the filter intensity threshold, and the intensity threshold is kept in as the 5 point into e to the power 3. Charge state, we are observing multiple charge states, so it is around 2 to 6. Dynamic exclusion, like exclude after n times, is 1. Exclusion duration is 40. Mass tolerance is we have set into ppm, not into m by z. And low and high is both as default parameters kept as 10. In MSMS mode, we have kept an isolation period of 2. We are using activation type as ACD. You can use CID and ETD depending upon your experiment. And we are using a collision energy mode as fixed. You can also step as stepped or assisted. Finally, we are using a detector type of ion trap. We are using a detector type of orbit trap. You can use the ion trap also. And the orbit trap resolution we have kept into 1500. Simultaneously, it is for the once the ion has been so fragmented. So we are keeping as 15,000. 15,000 in the detector type 2, which can be finally gone to, which you can include these parameters depending upon your molecule size. Okay, so once this is set, you can generate the method history. So once you are going for publication and all that, you can print this. You will know, like you can print this. Now you can know like whatever the parameters you have used in the document. So once this is set, we will incorporate this into the method. So you can see sample method have been lined up, and this is how the sample acquisition will begin. So this is from 0 to 120 minutes. Here we are increasing from 5% of B to 90% of the B, and you can see by the time it goes, the peptides are coming out, eluting from the column. You can monitor it on the real-time scale. You can see the peptides which are coming out, and you can also observe it remotely, like what is the quality.
All right. So what you see here at the end, which is what is shown here, which is the chromatogram of peptides. This is what you get from any given sample, right? So in our case study, which today we are talking about the let's say COVID-19. So when we took the nasopharyngeal swab sample from the human, these are from the respiratory specimens of individuals which were COVID suspected positive. Now, these kind of chromatograms were generated, right? And now from these chromatograms, one need to find out that what these proteins are. And at that stage, you need to do database search. So I hope now you are able to connect that first you have to prepare the samples, which requires protein quantification, peptide quantification. You need to then make a lot of settings changes in the LC and the MS and MS parameter. And then you can use different instrumentation like orbitrap fusion, which we show today, for separating your complex peptides. At the end, after doing all that entire complex work, then only you can get these kind of peptide bushes, which you need to search further and try to identify the which organism these proteins are coming from, what kind of protein and peptide sequences we have. So in this case of COVID-19 or from the nasopharyngeal swab sample, we were able to get many unique peptides of virus from different type of organic solvent which we use for the extraction. And then idea was, can we now take some of these peptides forward and develop a simplified test using another mass spectrometer, which is triple quadrupole based mass spectrometer known as target proteomics. So in the next part, we are going to show you demonstration for two instruments, two other mass spectrometers. One is cubicative Orbitrap and second is the TSQ Altis. Now, the cubicative mass spectrometer we actually used during the COVID time for metabolomics analysis of these samples for looking at the COVID-19 plasma, how that is showing any change in the metabolite based on the virus and host interaction which is happening in the human body. And further, the ALTIS was used for developing the target assays. So now I'm requesting our MSTAs to take the demonstration for QE forward, followed by ALTIS. So over to you, Avinash. Your audio has to be better, Avinash.
Is- okay.
Audible.
Should I repeat it? So this is another mass spec instrument. So Q exactly. So this has basically the two mass analyzers. So one is the quadrupole and another is like an orbitrap. So this both analyzer basically this instrument is very good for detecting the small molecules and other drugs from the different clinical samples. So here we are using this instrument for metabolomic studies and the small molecule screening and the microservice testing and the different kind of small molecule detection. So this is the UHPLC. So here we are taking our sample. So this unit have a different compartment. So this is the sample compartment. So where we kept our samples. So basically we kept the samples in the different areas. And after that, so these samples now this going to the different tubings and that is going to the mass spec for the analysis. This HPLC have a different unit like a pump which help in the transporting the solvent and the other samples to the mass spec. And this is a sample auto sampler where we kept our samples. And this is the column compartment, so which having like a columns from those samples are passing. And this is the UV detector compartment, so where we detect like a different molecule in the while passing, giving the different UV of the spectra and absorbing the spectra of the particular small molecule. And after that, this from this compartment, this is going to the mass spec, and from the mass spec, all the analysis will be happen here. So there are yeah. So just
So here you can see that. So this is the column, analytical column, where the sample is passing. So all the sample is passing through this kind of compartment and now going to the from the tubing to the next compartment. So if you see, like there are the different tubings, so which is taking the sample from this vials, and from this tubing it is going to that column, and now from the column it is going to the instrument. So here, so I'm just showing you a few chromatogram. So here we are trying to test some of the small molecule, which is a nitrosamine. So this is a different peak of different abundance peak of nitrosamine, and so we are detecting this nitrosamine at a different time point, and then on the basis of the retention time. So this is the abundance of each peak, and this is one of the molecules. Same way we are detecting the different small molecules and other metabolites from the this instrument. So yeah. Oh.
All right, Avinash, thank you. So let's now move on to TSQ test quickly for targeted proteomics based demonstration. So Shashwati.
Yeah. So this is the TSQ ortiz. So as the name suggested, it is the triple quadruple instrument which has two quadruple and one collagen cell. So it has three main compartments. So first is you can see the solvent system here, and from this solvent system, the solvent goes into the pump. So here we have all the pump system. From all the four systems, the samples came here. Like it has a quaternary pump. After that, this is the auto sampler. So here we keep our sample. So it has many trays. So we keep our sample here to the analysis. After that, from the sample, here is the column compartment. So here the sample comes the column compartment. Yeah. So this is the column. Then from this column, the sample directly goes to the mass spectrometer. So here this is the voltage. So sample are coming directly from here to the mass spectrometer. Mainly TST voltage is used for targeted analysis. So in fusion, you already know that we do discovery programs. From those we pick some of the protein targets and we target those to validate our system. We do some of the run QC samples to show that day to day how these samples are varying in the instrument. So here it is the BSA protein which I am showing. It has different peptide output, and this is the peak showing, and the bar plot is showing the intensity of the BSA. So in this bar graph we monitor that how the instrument is performing day-wise. So for that we ran BSA. Also we have NCF7, which is a complex cell line sample. Also we monitor it as a QC sample. Here we have optimized different proteins and we monitor its intensity throughout day-wise to see how the instrument is performing. So mainly the application of this instrument is we can monitor let's say COVID positive and negative sample which we are seeing. Then we can monitor one protein that is coming by upregulated. So here I will show you one of the project that we did in the COVID time. So we inject the internal standard in our sample in both positive and negative sample to show there is no variation in the spike peptide. So here you can see COVID negative and positive, both the peptide here are showing the same label. But when we are going to different peptide, you can see this particular peptide, it is high in case of COVID positive, but it's not present in case of COVID negative. Similarly, for different peptides, we can see. So in positive, it is highly upregulated, but it's not present in the negative.
All right, great. Thank you, Shashwati and TS for giving the demonstration. So as we are just talking about COVID-19, and you just have seen this demonstration of triple quadruple mass spec. So finally, what we were able to look for, various peptides of virus that can be developed the as a simplified test in 8 minutes time of elution of these peptides based on the 11 peptides of virus from spike protein, nucleoprotein, and replicase polyprotein. And this is known as the targeted MRM assay to look for the specific peptides of interest rather than now relying on thousands to millions of peptides. We can now selectively look at peptides of our interest. But in the process, you need to again inject some known peptides for the quality control check as a spike in the internal standard. And sometime if you want to develop the quantitative assay, you need to do heavy label peptide for developing the quantitative assay. So these are various developments which are happening in the field, which can be taken forward. But I hope you got a complete workflow of starting from samples to developing an assay using mass spectrometry based approaches. So very quickly, let me request Dr. Arghya Banerjee to give a short demonstration of one of the software which can be used for the mass spectrometry data analysis, known as the Proteome Discoverer. There are actually many other software available as well. One which is gaining momentum right now is MS Fragger. Additionally, the MaxQuant is software. But Proteome Discoverer, we will take very short demonstration now. Arghya, you want to share your screen now? So while I see some delay over there, ideally, like, you are now talking about this pool of peptide which you have acquired the data from different mass spectrometers, and then the data mining becomes very crucial to really get an idea about which organism they belong to, what kind of protein we have identified. So this entire process of data analysis is actually what makes you a good scientist for doing proteomics. While you can really acquire the data very short time. For example, in this case, the nasopharyngeal data of proteome we could get in 2 hours time. But what you can generate 3 type of information. One is you are looking for the viral infection and the viral peptides from the given sample. So we first searched against SARS-CoV-2 database. Then additionally, we were also interested to know the host response. So we looked at the human proteins and human proteome database. And now more recently, we thought about what was the environment or microenvironment which was affecting the individuals. So we also looked at now the metaproteome of all the pathogenic databases. So think about the data acquisition was very straightforward, 2 hours. But now data analysis took several months time of a lot of computing power to make better sense of this data. So all right, Arghya, very quickly you can give overview of Proteome Discoverer.
So my voice is audible, correct?
Yep.
Okay, great. So this is one of the robust proprietary tool which is being owned by the Thermo. So Proteome Discoverer is one of the robust tool which we use. So I will just give you a brief. You can initiate a study by file operating into a new study. In the when you are doing a new study, you will be getting the workflows. The two important workflows are the processing and the consensus. So I will just add the processing and the consensus workflow. That maybe the discussion like how the processing and consensus workflow are set in some other time. So yeah, I have given the processing and consensus workflow. I will label my study here. Like in this study, I can do the labeling, and I simultaneously I can add the files. Like these are my QC files, like which can be added, or the SARS-CoV-2 files, and I will just press OK. And once these all things have been defined, like once the study is defined, I will just click at run. Like grouping and quantification workflow, I will just save and start at run. Once that has been done, like you will get a protein list of like this one, like you will get a protein list which will carry the accession ID, the description, the sequence, what is the sequence of the peptides which have been identified with the human FASTA. Then here it comes the coverage, like for each individual protein, what is the confidence, like what is the confidence of the coverage.
So if we go like this is very obvious, like if you go from higher to lower, if you are using the human tissue samples, the hemoglobin has to be there because of the blood samples and all. So once that is done, you can also do the what kind of modification it can adapt, like whether it is a methionine loss or whether it is a acetylation or a phospho. Yeah, so once this is done, you can do a filtration, like you want to select only those proteins which are like very high much confident. In a basic thumb rule which we generally follow in our proteomics lab at IIT Bombay, we will take only those unique peptides, and this is also recommended by the standard guidelines of the proteomics. We will take only those proteins which have a unique peptides of 2 or greater than 2. Once that is done, we will go for the protein FDR confidence, which at least have to be high. So once this is done, you just do the filtration and you will get the list of the proteins. Also, the data can be identified from here, like you will find something around proteins, protein groups, PSMs and isoforms. Just I come to the now the visualization of the data. So yeah, in the visualization of the data, so first of all, how you know it is a good or a bad data, whether the abundances like whether the sample run has been optimized or not. So this is the form. So I will just go one by one. So this is how it looks like so you can see these are the samples or the COVID samples which have been used, and yes, the samples are quite well normalized. The normalizations is looking good. You can go and check the PCAs also. You can go and check the volcanoes plot also, as well as the heat map. I will not go in that thing as a much more detail, but yeah, you can do all kinds of work in this. You can go for the bar chart. So one of the important parameters we monitor the miscleavages. So yeah, if your miscleavage is more than 89.5%, like zero miscleavages, that means what your digestion was perfect. So like these kind of every datas can be pulled out, every modification datas, like whether what kind of data you want to use, it can be pulled out, it can be churned, and after that you can go for the biostatistical analysis for making it more visual or more interactive. So this is one of the method in which the instrument is being used. Now, like yes, Professor, you can take it back, and after that we can.
All
Yeah.
right, great. So thank you for giving a very quick and crisp overview of Proteome Discoverer. There are many software, as you mentioned, and as a scientist, the major challenge is to really, analyze this complex proteomic data sets. So in the last minute or so, let me just, share an interesting question which I had received from a student. The student asked me from the MOOC course that we emphasize so much about mass spectrometry technologies, uh, but what are the career options or the possibilities in this area if we are trying to build our career in this particular space? So see, like the mass spectrometers are currently still, I would say, like really industry requirements, and there is a dearth of trained individuals in this area. If you are well trained in the good instrumentation of mass spectrometers, and more importantly, good data analysis skill, you will be in great demand everywhere. And especially even right now in California, I see there is a huge requirements and huge demand of people trained in this particular area. So, of course, if your interest takes you further, this is something where you can really try to take your training forward and see how best you can understand the complex proteome of different biological organism, different type of complex samples. The same instruments are also being used actively for various type of biotherapeutics and pharmaceutical applications. And that's where you can see that mass spectrometers are just not about core proteomics, but they have the wider applications, including in metabolomics as well as in the pharmacological and pharmaceutical sciences as well. So in today's live session of mass spectrometry-based proteomics, I hope you got a flavor of different steps involved, starting from the key concept, the sample prep, to moving to the quantification, then looking at the instrumentation, method settings, and finally the data analysis. Of course, there's a lot which we can do in this area, but aim was to make it more lively demonstration for you and give you the flavor of these technologies from our lab. I hope you have enjoyed The MOOC course so far, the bioengineering students, I think good luck for your exams. And the Interactomics students, I think the last few weeks which you have, you will be also exposed to new technologies. And our next live session in the April is going to cover about more of the biosensors and the array-based technologies of proteomics to give you the flavor of that directly from our lab. So thanks everyone for joining, and especially all the TAs for keeping it very, uh, good live demonstration in a very short time. So thanks the entire team, and we look forward to see you again next month. Thank you everyone.